Chapter 1: Understanding Life, the Foundations of Biology. Biology stands as one of the most fascinating and rapidly evolving scientific disciplines, encompassing the study of all living organisms from the smallest bacteria to the largest mammals. This comprehensive guide will take you through the essential concepts that form the foundation of biological understanding, providing you with the tools to appreciate the complexity and beauty of life on Earth. Whether you are a student beginning your journey in biology or someone seeking to deepen your understanding of the natural world, this guide will serve as your roadmap through the interconnected realms of biological science. The study of biology begins with recognizing what distinguishes living organisms from non-living matter. Life exhibits several fundamental characteristics that set it apart from the inanimate world. Organization represents the first hallmark of life, as all living things display a high degree of structural complexity and order. From the molecular level to entire organisms, life maintains intricate arrangements of components that work together harmoniously. This organization extends beyond individual organisms to populations, communities, and entire ecosystems. Metabolism encompasses all the chemical reactions that occur within living organisms to maintain life. These reactions include both catabolic processes that break down complex molecules to release energy, and anabolic processes that use energy to build complex molecules from simpler components. Metabolism enables organisms to obtain energy from their environment, convert it into usable forms, and use it to power all life processes. The remarkable efficiency and precision of metabolic pathways demonstrate the sophisticated chemistry that underlies all biological functions. Growth and development represent another defining characteristic of life, as organisms increase in size and complexity over time, according to genetic instructions. Growth involves an increase in the number and size of cells, while development encompasses the progressive changes that transform a simple fertilized egg into a complex multicellular organism. These processes require precise coordination of cellular activities and demonstrate the remarkable ability of genetic information to guide the formation of complex structures. Reproduction ensures the continuation of life by enabling organisms to produce offspring that carry genetic information into future generations. Sexual reproduction combines genetic material from two parents, creating offspring with unique genetic combinations that provide variation for evolutionary adaptation. Asexual reproduction produces genetically identical offspring, allowing successful genetic combinations to be perpetuated efficiently. The diversity of reproductive strategies reflects the various solutions that evolution has produced for the fundamental challenge of species survival. Response to stimuli enables organisms to detect and react to changes in their environment, a capability essential for survival in a dynamic world. Plants grow toward light sources, animals flee from predators, and bacteria move toward nutrients. These responses require sophisticated sensory systems that can detect environmental changes, and communication systems that can coordinate appropriate reactions. The ability to respond appropriately to environmental stimuli demonstrates the integration of structure and function that characterizes living systems. Homeostasis refers to the maintenance of stable internal conditions despite changes in the external environment. Living organisms must regulate factors such as temperature, pH, water balance, and nutrient concentrations within narrow ranges to ensure proper cellular function. Homeostatic mechanisms involve feedback systems that detect deviations from optimal conditions and initiate corrective responses. This dynamic equilibrium requires constant energy expenditure, but enables organisms to survive in variable environments. Evolution through natural selection provides the overarching framework that explains both the unity and diversity of life. All organisms share common ancestry, yet millions of species have evolved to exploit different environmental opportunities. Natural selection favors individuals with traits that enhance survival and reproduction, leading to evolutionary changes over time. This process has produced the remarkable diversity of life forms we observe today, while maintaining the fundamental biochemical and cellular processes that unite all living things. The scientific method provides the foundation for biological research, ensuring that our understanding of life rests on empirical evidence rather than speculation. Scientists formulate hypotheses based on observations, design experiments to test these hypotheses, collect and analyze data, and draw conclusions supported by evidence. This systematic approach has led to tremendous advances in biological knowledge and continues to drive new discoveries about the nature of life. The levels of biological organization create a hierarchy that extends from molecules to the biosphere, with each level exhibiting properties that emerge from the interactions of components at lower levels. Atoms combine to form molecules, molecules organize into cellular structures, cells aggregate into tissues, tissues combine to form organs, organs work together in organ systems, and organ systems comprise individual organisms. These organisms interact with others to form populations, communities, and ecosystems that ultimately constitute the biosphere. Modern biology has been revolutionized by technological advances that enable scientists to study life at unprecedented scales of detail and complexity. Microscopy reveals the intricate structures of cells and their components, while DNA sequencing technologies allow scientists to read the genetic instructions that guide life processes. Computer modeling helps researchers understand complex biological systems, while genetic engineering techniques enable the modification of organisms for research and practical applications. The interdisciplinary nature of modern biology reflects the complexity of living systems and the need for diverse approaches to understand them fully. Biochemistry examines the chemical processes within organisms, while molecular biology focuses on the structure and function of biological molecules. Genetics studies heredity and variation. Ecology investigates relationships between organisms and their environment, and evolutionary biology examines how species change over time. These and other subdisciplines contribute to our comprehensive understanding of life. The practical applications of biological knowledge extend far beyond academic interest, influencing medicine, agriculture, biotechnology, and environmental management. Medical advances rely on understanding how the human body works and how diseases disrupt normal functions. Agricultural improvements depend on knowledge of plant and animal biology, genetics, and ecology. Biotechnology harnesses biological processes to produce useful products and services. Environmental management requires understanding ecological principles and the impacts of human activities on natural systems. Conservation biology applies biological principles to protect biodiversity and preserve natural ecosystems for future generations. As human activities increasingly impact the natural world, understanding biological processes becomes essential for developing sustainable practices that can meet human needs while protecting the environment. The study of biology thus serves both intellectual curiosity and practical necessity, providing the knowledge needed to address some of humanity's greatest challenges. The historical development of biological knowledge reveals how our understanding of life has evolved through careful observation, experimentation, and theoretical development. Early naturalists documented the diversity of life and proposed classification systems, while later researchers discovered the cellular basis of life and the molecular mechanisms that control biological processes. The integration of these discoveries with evolutionary theory created the modern synthesis that unifies all of biology under common principles. As we embark on this journey through biology, remember that living systems exhibit remarkable complexity that emerges from relatively simple underlying principles. The same chemical and physical laws that govern non-living matter also apply to biological systems, yet the organization and interactions of biological components create properties and behaviors that transcend simple chemistry and physics. Understanding these emergent properties requires thinking about biological systems at multiple levels simultaneously, appreciating how molecular interactions give rise to cellular functions, how cellular activities produce organism-level behaviors, and how organism interactions create ecosystem-level patterns. Chapter 2. molecular biology and cellular structure. The foundation of all life rests on the intricate dance of molecules within cells, where complex chemical processes transform simple raw materials into the sophisticated machinery of living organisms. Understanding molecular biology and cellular structure provides the key to comprehending how life functions at its most fundamental level. This chapter will guide you through the essential molecules that constitute living matter and the cellular structures that organize these molecules into functional units capable of maintaining, reproducing, and evolving life. Water serves as the universal solvent of life, creating the aqueous environment in which all biological processes occur. The unique properties of water arise from its polar structure, which enables it to form hydrogen bonds with other water molecules and with many biological compounds. These hydrogen bonds give water its high specific heat capacity, allowing it to moderate temperature changes in living organisms. Water's cohesive properties enable it to move through plants from roots to leaves, while its ability to dissolve many substances makes it an ideal medium for transporting nutrients and waste products throughout organisms. Carbohydrates provide energy and structural support for living organisms, ranging from simple sugars that fuel cellular processes to complex polysaccharides that form structural components. Monosaccharides such as glucose serve as primary energy sources, while disaccharides like sucrose transport energy in plants. Polysaccharides perform diverse functions. Starch stores energy in plants, glycogen stores energy in animals, cellulose provides structural support in plant cell walls, and chitin forms the exoskeletons of arthropods. The chemical structure of carbohydrates, built around carbon atoms bonded to hydrogen and oxygen, reflects their role as energy storage and structural molecules. Lipids constitute a diverse group of biological molecules that includes fats, oils, phospholipids, and steroids, all characterized by their hydrophobic nature. Triglycerides store energy more efficiently than carbohydrates, providing twice as much energy per gram. Phospholipids form the structural basis of cellular membranes, with their hydrophilic heads and hydrophobic tails creating the bilayer structure that separates cellular compartments. Steroids, such as cholesterol, maintain membrane fluidity and serve as precursors for important signaling molecules, including hormones. The hydrophobic nature of lipids enables them to form barriers that separate aqueous compartments while storing concentrated energy. Proteins represent the most structurally and functionally diverse molecules in living organisms, performing roles that include catalysis, structure, transport, defense, regulation, and movement. The primary structure of proteins consists of chains of amino acids linked by peptide bonds, with the sequence of amino acids determined by genetic information. Secondary structure includes regular patterns such as alpha helices and beta sheets stabilized by hydrogen bonds. Tertiary structure results from interactions between amino acid side chains, creating the three-dimensional shape that determines protein function. Quaternary structure involves the association of multiple protein chains into functional complexes. Enzymes represent specialized proteins that catalyze the chemical reactions essential for life, lowering activation energy barriers and enabling reactions to proceed at rates compatible with biological processes. The active site of an enzyme provides a precisely shaped environment that binds specific substrates and facilitates their conversion to products. Enzyme regulation through allosteric effects, competitive inhibition, and covalent modification allows cells to control metabolic pathways and respond to changing conditions. Understanding enzyme function is crucial for comprehending how cells manage the thousands of chemical reactions that sustain life. Nucleic acids store and transmit genetic information, with DNA serving as the repository of hereditary instructions, and RNA playing diverse roles in gene expression and regulation. DNA consists of two complementary strands wound together in a double helix, with the sequence of bases encoding genetic information. RNA typically exists as single strands that can fold into complex structures, enabling functions such as protein synthesis, gene regulation, and catalysis. The central dogma describes the flow of genetic information from DNA to RNA to proteins, though we now know that RNA also plays regulatory roles that modify this simple linear flow. The cell membrane forms the boundary that separates the internal environment of cells from their external surroundings, while controlling the passage of materials in and out of cells. The fluid mosaic model describes membranes as dynamic structures composed of phospholipid bilayers embedded with proteins that move laterally within the membrane. Membrane proteins serve diverse functions including transport, signal reception, and enzymatic activity. The selective permeability of membranes enables cells to maintain internal concentrations of substances that differ from external concentrations, creating the chemical gradients essential for many cellular processes. Prokaryotic cells, including bacteria and archaea, represent the simplest and most ancient forms of cellular life, lacking membrane-bound organelles and nuclei. Despite their apparent simplicity, prokaryotic cells exhibit remarkable diversity and sophistication, with specialized structures such as flagella for movement, pili for attachment and genetic exchange, and various internal membrane systems for specific functions. The cell wall provides structural support and protection, while the plasma membrane controls material transport. The nucleoid region contains genetic material that is not separated from the cytoplasm by a membrane, allowing direct interaction between DNA and the protein synthesis machinery. Eukaryotic cells contain membrane-bound organelles that compartmentalize cellular functions, enabling greater complexity and specialization than is possible in prokaryotic cells. The nucleus houses genetic material and controls cellular activities through the regulation of gene expression. The endomembrane system, including the endoplasmic reticulum and Golgi apparatus, processes and transports proteins and lipids throughout the cell. Lysosomes digest worn-out cellular components and foreign materials, while peroxisomes break down toxic substances and participate in lipid metabolism. Mitochondria serve as the powerhouses of eukaryotic cells, converting the chemical energy stored in organic molecules into ATP through the process of cellular respiration. The double membrane structure of mitochondria creates specialized compartments for different stages of respiration, with the inner membrane containing the electron transport chain that generates the proton gradient driving ATP synthesis. Mitochondria contain their own DNA and ribosomes, supporting the endosymbiotic theory that these organelles evolved from ancient bacterial cells that were engulfed by early eukaryotic cells. Chloroplasts enable plants and algae to capture light energy and convert it into chemical energy through photosynthesis, making them the ultimate source of energy for most life on Earth. The thylakoid membranes contain chlorophyll and other pigments that absorb light energy, while the surrounding stroma contains enzymes for carbon fixation. Like mitochondria, chloroplasts have their own genetic material and are thought to have evolved from endosymbiotic cyanobacteria. The coordination between chloroplasts and mitochondria allows plants to both capture energy through photosynthesis and release it through respiration. The cytoskeleton provides structural support and organization to eukaryotic cells while enabling cellular movement and organelle transport. Microfilaments composed of actin provide mechanical support and enable cellular movement through contraction and extension. Intermediate filaments offer tensile strength and help maintain cell shape. Microtubules serve as tracks for organelle transport and form the spindle apparatus that separates chromosomes during cell division. The dynamic nature of the cytoskeleton allows cells to change shape, move, and reorganize their internal components in response to internal and external signals. Cellular transport mechanisms enable cells to move materials across membranes and throughout their interior, maintaining the concentration gradients and molecular distributions essential for cellular function. Passive transport processes, including diffusion and osmosis, move materials down concentration gradients without energy expenditure. Active transport uses cellular energy to move materials against concentration gradients, often through specialized protein pumps. Bulk transport processes such as endocytosis and exocytosis enable cells to engulf large particles or secrete materials in membrane-bound vesicles. Cell communication allows multicellular organisms to coordinate their activities and respond to environmental changes through complex signaling networks. Chemical signals, including hormones, neurotransmitters, and growth factors, bind to specific receptor proteins, triggering intracellular signaling cascades that amplify and integrate signals. These pathways enable cells to respond appropriately to multiple simultaneous signals and coordinate their activities with other cells. Understanding cellular communication is essential for comprehending how multicellular organisms function as integrated units. The integration of molecular and cellular processes creates the emergent properties that characterize living systems, demonstrating how complex behaviors arise from the interactions of simpler components. Metabolism emerges from the coordinated activities of thousands of enzymes. Growth results from the controlled synthesis and assembly of cellular components, and reproduction requires the precise duplication and distribution of genetic material. This integration occurs at multiple levels simultaneously, with molecular interactions creating cellular functions, cellular activities producing tissue-level behaviors, and tissue coordination enabling organism-level responses. Chapter 3: Genetics and Heredity. Genetics illuminates one of biology's most profound mysteries: how traits pass from parents to offspring, and how genetic information shapes the characteristics of living organisms. This field reveals the molecular mechanisms underlying heredity, while explaining the variation that enables species to evolve and adapt to changing environments. Understanding genetics provides insights into development, disease, evolution, and the fundamental processes that perpetuate life across generations. The discovery of inheritance patterns began with Gregor Mendel's systematic experiments with pea plants in the mid-19th century. Mendel's careful observations revealed that traits are controlled by discrete factors, now known as genes, that exist in different versions called alleles. His law of segregation states that allele pairs separate during gamete formation, with each gamete receiving only one allele for each gene. The law of independent assortment describes how genes for different traits are inherited independently, though we now know this applies only to genes located on different chromosomes or far apart on the same chromosome. The molecular basis of heredity resides in DNA, the remarkable double-stranded molecule that stores genetic information in all living organisms. The structure of DNA, with its complementary base pairs and anti-parallel strands, enables both stable storage of genetic information and accurate replication during cell division. The sequence of bases along DNA strands constitutes a genetic code that specifies the amino acid sequences of proteins, the molecules that carry out most cellular functions. This elegant system links genotype, the genetic constitution of an organism, with phenotype, the observable characteristics that result from genetic and environmental interactions. Chromosomes organize genetic material within cells and ensure its accurate transmission during reproduction. Most organisms possess paired sets of chromosomes, with one member of each pair inherited from each parent. Humans have 23 pairs of chromosomes, including 22 pairs of autosomes and one pair of sex chromosomes that determine biological sex. During meiosis, the specialized cell division that produces gametes, chromosome pairs separate and genetic material is exchanged between homologous chromosomes, creating genetic diversity in offspring. Gene expression involves the complex process by which genetic information is converted into functional proteins through transcription and translation. Transcription produces RNA copies of genes, while translation uses these RNA molecules as templates to synthesize proteins. Gene regulation controls when and where genes are expressed, enabling cells to respond to developmental signals and environmental changes while maintaining their specialized functions. Epigenetic modifications can alter gene expression without changing DNA sequences, providing additional layers of genetic control. Inheritance patterns extend beyond simple Mendelian genetics to include phenomena such as incomplete dominance, codominance, and polygenic inheritance. Incomplete dominance produces intermediate phenotypes in heterozygotes, while codominance results in the simultaneous expression of both alleles. Polygenic traits are controlled by multiple genes, each contributing to the overall phenotype, resulting in continuous variation rather than discrete categories. Environmental factors also influence gene expression, creating complex interactions between genetics and environment in determining phenotypes. Mutations represent changes in DNA sequences that provide the ultimate source of genetic variation underlying evolution. Point mutations involve changes in single nucleotides, while larger mutations can include insertions, deletions, or rearrangements of DNA sequences. Some mutations have no effect on organism function, others can be beneficial or harmful. Spontaneous mutations arise from errors in DNA replication or damage from environmental factors, while induced mutations result from exposure to mutagens such as radiation or chemicals. Genetic disorders demonstrate the importance of normal gene function and illustrate how mutations can cause disease. Single gene disorders such as sickle cell anemia and Huntington's disease result from mutations in individual genes with large effects. Chromosomal disorders like Down syndrome involve abnormalities in chromosome number or structure. Complex genetic disorders involve multiple genes and environmental factors, making them more difficult to predict and treat. Understanding the genetic basis of disease has revolutionized medicine by enabling improved diagnosis, treatment, and prevention. Population genetics examines genetic variation within and between populations, providing insights into evolution, conservation, and human health. Allele frequencies describe the prevalence of different gene versions within populations, while the Hardy-Weinberg principle provides a mathematical framework for understanding how these frequencies change over time. Factors such as mutation, migration, genetic drift, and natural selection all influence population genetic structure and can lead to evolutionary change. Modern genetic technologies have revolutionized our ability to study and manipulate genes, opening new frontiers in research and practical applications. DNA sequencing enables scientists to read genetic information quickly and accurately, while genetic engineering techniques allow precise modification of DNA sequences. Gene therapy offers potential treatments for genetic diseases by introducing functional genes into patient cells. Genetic testing can identify disease predispositions and guide medical decisions, while pharmacogenomics uses genetic information to optimize drug treatments. The Human Genome Project, completed in 2003, provided the first complete sequence of human DNA and demonstrated the power of large-scale genomic approaches. This international effort revealed that humans have fewer genes than expected, and that large portions of the genome do not encode proteins. Comparative genomics shows that humans share most of their genes with other species, reflecting common evolutionary ancestry. Personal genomics now enables individuals to access their own genetic information for medical and ancestry applications. Genetic counseling helps individuals and families understand genetic information and make informed decisions about reproduction and medical care. Genetic counselors assess genetic risks, explain test results, and provide support for decision-making. Prenatal genetic testing can identify genetic disorders before birth, while pre-implantation genetic diagnosis enables selection of embryos without genetic diseases during in vitro fertilization. These applications raise important ethical questions about the appropriate use of genetic information. The relationship between genes and development reveals how genetic information guides the transformation of single cells into complex multicellular organisms. Developmental biology shows how gene expression patterns create different cell types and organize them into tissues and organs. Homeotic genes control body plan development, while growth factors coordinate cellular activities during development. Understanding development provides insights into birth defects, cancer, and regenerative medicine. Evolutionary genetics examines how genetic changes drive evolutionary processes and create the diversity of life on Earth. Natural selection acts on genetic variation to favor alleles that enhance survival and reproduction. Genetic drift causes random changes in allele frequencies, particularly in small populations. Gene flow between populations can introduce new alleles or homogenize genetic differences. Molecular evolution tracks genetic changes over time and reveals evolutionary relationships between species. Behavioral genetics investigates the genetic basis of animal behavior, revealing how genes influence everything from basic reflexes to complex social behaviors. While most behaviors result from interactions between multiple genes and environmental factors, some genetic variants have clear behavioral effects. Understanding behavioral genetics has implications for human psychology, mental health, and social policy, though it also raises concerns about genetic determinism and discrimination. Agricultural genetics applies genetic principles to improve crop plants and livestock, addressing the challenge of feeding a growing global population while maintaining environmental sustainability. Plant breeding programs use genetic knowledge to develop varieties with improved yield, disease resistance, and nutritional content. Animal breeding applies similar principles to livestock improvement. Genetic engineering enables the introduction of genes from other species, creating transgenic organisms with novel characteristics. The ethical implications of genetic research and technology continue to evolve as our capabilities expand. Issues include genetic privacy and discrimination, the use of genetic information in criminal justice, and the potential for genetic enhancement of human capabilities. Gene editing technologies raise questions about modifying human embryos and the potential consequences for future generations. Public engagement and thoughtful regulation are essential for ensuring that genetic technologies benefit society while respecting individual rights and values. As genetic technologies become more powerful and accessible, their impact on society will continue to grow. Precision medicine uses genetic information to tailor treatments to individual patients, while synthetic biology seeks to engineer biological systems with novel functions. Understanding genetics will be essential for addressing challenges such as climate change, food security, and emerging diseases. The future of genetics promises continued discoveries that will deepen our understanding of life, while raising new questions about the responsible use of genetic knowledge. Chapter 4: Evolution and the Diversity of Life. Evolution stands as biology's greatest unifying principle, providing a comprehensive framework for understanding the history and diversity of life on Earth. This powerful scientific theory explains how all organisms share common ancestry, while accounting for the remarkable variety of forms, functions, and behaviors we observe in nature. Understanding evolution is essential for comprehending not only the past history of life, but also its future trajectory in response to changing environmental conditions. The theory of evolution by natural selection emerged in the 19th century through the independent work of Charles Darwin and Alfred Russel Wallace, revolutionizing our understanding of life's diversity. Darwin's meticulous observations during his voyage on HMS Beagle, particularly his studies of finches in the Galapagos Islands, revealed patterns suggesting that species could change over time. His subsequent formulation of natural selection provided a testable mechanism for evolutionary change, establishing evolution as a scientific theory supported by evidence from multiple disciplines. Natural selection operates through differential reproductive success, favoring individuals with traits that enhance survival and reproduction in specific environments. This process requires three essential conditions: variation in traits among individuals, heritability of those traits, and differential reproductive success associated with trait variation. When these conditions are met, favorable traits become more common in populations over time, while unfavorable traits become rarer. Natural selection thus shapes populations to become better adapted to their environments through gradual change. The evidence for evolution comes from diverse sources that independently support the same conclusion: life has evolved over time from common ancestors. The fossil record documents the history of life, showing how organisms have changed through geological time and revealing transitional forms that bridge major evolutionary transitions. Comparative anatomy reveals homologous structures that suggest common ancestry, while embryological similarities between species provide evidence for shared developmental pathways. Molecular evidence, including DNA and protein sequences, allows precise reconstruction of evolutionary relationships. Mechanisms of evolution extend beyond natural selection to include genetic drift, gene flow, and mutation, each contributing to evolutionary change in different ways. Genetic drift represents random changes in allele frequencies that occur in all populations, but have greater effects in smaller populations. Gene flow involves the movement of alleles between populations through migration, which can introduce new genetic variation or homogenize differences between populations. Mutation provides the ultimate source of genetic variation by creating new alleles, though most mutations are neutral or harmful rather than beneficial. Speciation, the process by which new species arise, occurs when populations become reproductively isolated and diverge genetically over time. Geographic isolation often initiates speciation by preventing gene flow between populations, allowing them to evolve independently in response to local conditions. Accumulated genetic differences can eventually lead to reproductive incompatibility, completing the speciation process. Adaptive radiation occurs when a single ancestral species gives rise to multiple descendant species adapted to different ecological niches, dramatically increasing biodiversity. The history of life on Earth spans approximately 3.8 billion years, revealing major evolutionary transitions that have shaped the biosphere. The origin of life marked the beginning of biological evolution, though the precise mechanisms remain uncertain. The evolution of photosynthesis transformed Earth's atmosphere by producing oxygen, enabling the evolution of aerobic respiration and complex multicellular life. Mass extinction events have periodically eliminated large percentages of species, creating opportunities for surviving lineages to diversify and fill vacant ecological niches. Phylogenetics reconstructs evolutionary relationships between organisms using morphological, molecular, and other evidence. Phylogenetic trees represent hypotheses about evolutionary relationships, with branch points indicating common ancestors and branch lengths often representing evolutionary time or genetic change. These evolutionary trees reveal patterns that inform our understanding of how different groups of organisms are related and when major evolutionary events occurred. Modern phylogenetics relies heavily on molecular data, which provides unprecedented resolution of evolutionary relationships. Adaptation represents the fit between organisms and their environments that results from natural selection, acting on heritable variation. Adaptations can be morphological structures, physiological processes, or behavioral patterns that enhance survival and reproduction in specific environments. Examples include the streamlined body shape of aquatic animals, the thick fur of Arctic mammals, and the complex social behaviors of many species. Understanding adaptation helps explain the remarkable diversity of solutions that evolution has produced for environmental challenges. Convergent evolution demonstrates how similar environmental pressures can produce similar adaptations in unrelated organisms, revealing the power of natural selection to find effective solutions to environmental challenges. The evolution of flight in birds, bats, and insects represents convergent evolution, as does the development of echolocation in dolphins and bats. These examples show that evolution can independently arrive at similar solutions, supporting the idea that natural selection shapes organisms to fit their environments. Sexual selection represents a special form of natural selection that operates through differential mating success rather than survival differences. Traits that enhance an individual's ability to attract mates or compete for mates can evolve even if they reduce survival prospects. Elaborate plumage in male birds, antlers in deer, and complex courtship behaviors all represent products of sexual selection. This process helps explain some of the most striking and seemingly impractical features of organisms, while contributing to reproductive isolation and speciation. Coevolution occurs when two or more species evolve in response to each other, creating reciprocal evolutionary pressures that can lead to remarkable adaptations. Predator-prey relationships often involve coevolutionary arms races, where predators evolve more effective hunting strategies while prey evolve better defenses. Plant-pollinator relationships represent mutualistic coevolution, where both partners benefit from their interaction and evolve traits that enhance the relationship. Coevolution helps explain the intricate relationships between species and the complexity of ecological communities. Human evolution illustrates how evolutionary principles apply to our own species and reveals our place in the tree of life. Humans share common ancestors with other primates, and fossil evidence documents the gradual evolution of human characteristics, including bipedalism, enlarged brains, and tool use. Genetic studies reveal that humans are remarkably similar to other primates at the molecular level, while also showing how recent human evolution has occurred in response to cultural and environmental changes. Understanding human evolution provides insights into human nature and our relationship with other species. The impact of human activities on evolution includes both direct effects through selective pressures and indirect effects through environmental changes. Overharvesting can cause rapid evolutionary changes, as seen in the evolution of smaller body sizes in heavily exploited fish populations. Pollution creates strong selective pressures that can lead to rapid adaptation, while habitat fragmentation reduces gene flow and increases genetic drift. Antibiotic use has driven the evolution of resistant bacteria, demonstrating evolution in action and highlighting the importance of evolutionary thinking in medicine. Conservation biology applies evolutionary principles to protect biodiversity and prevent extinctions. Understanding evolutionary relationships helps identify species that represent unique evolutionary heritage and prioritize conservation efforts. Genetic diversity within species is important for their evolutionary potential and ability to adapt to environmental changes. Maintaining large population sizes and connectivity between populations helps preserve evolutionary processes and enable species to respond to future environmental challenges. Evolutionary medicine applies evolutionary principles to understand human health and disease, recognizing that many health problems result from mismatches between our evolutionary heritage and modern environments. Many diseases represent evolutionary trade-offs or result from pathogens that evolve rapidly to overcome our defenses. Understanding the evolutionary history of pathogens helps predict their evolution and guides treatment strategies. Evolutionary approaches to medicine are providing new insights into cancer, infectious diseases, and mental health. The study of evolution continues to generate new insights as technological advances provide new tools for investigating evolutionary processes. Experimental evolution allows scientists to observe evolutionary changes in real time under controlled conditions. Ancient DNA studies provide direct evidence of evolutionary changes over time. Mathematical models are becoming increasingly sophisticated in their ability to predict evolutionary outcomes. These advances demonstrate that evolution remains an active and vibrant field of research, with practical applications for addressing human challenges. Understanding evolution is essential for addressing many contemporary challenges, including climate change, emerging diseases, food security, and biodiversity conservation. As we face an uncertain future, evolutionary principles provide guidance for understanding how life might respond to changing conditions and how we can work to preserve the evolutionary processes that have created the magnificent diversity of life on Earth. Evolution not only explains the past history of life, but also provides the framework for understanding its future trajectory. Chapter 5: Ecology and Environmental Interactions. Ecology reveals the intricate web of relationships that connect all living organisms with each other and with their physical environment, demonstrating that no organism exists in isolation. This field of biology examines how organisms interact within communities, how energy flows through ecosystems, and how environmental factors shape the distribution and abundance of life on Earth. Understanding ecological principles is crucial for comprehending the complexity of natural systems and for addressing the environmental challenges facing our planet. The hierarchical organization of ecological systems provides a framework for understanding nature at multiple scales, from individual organisms to the entire biosphere. Individual organisms represent the basic units of ecology, each adapted to survive and reproduce in specific environments. Populations consist of individuals of the same species living in the same area, exhibiting characteristics such as size, density, and age structure that influence their dynamics and interactions with other populations. Communities encompass all the populations of different species, that live together in a particular area and interact with one another. Ecosystems include both the living communities and their physical environment, emphasizing the flow of energy and cycling of nutrients through the system. Biomes represent large-scale ecosystems characterized by similar climate conditions and dominant vegetation types, such as tropical rainforests, temperate grasslands, and Arctic tundra. The biosphere encompasses all of Earth's ecosystems and represents the global sum of all life and the environments that support it. Understanding these different levels of organization helps ecologists study patterns and processes at appropriate scales. Energy flow through ecosystems follows fundamental physical laws and creates the foundation for all ecological relationships. Primary producers, primarily green plants and other photosynthetic organisms, capture energy from sunlight and convert it into chemical energy stored in organic molecules. This energy flows through food webs as primary consumers feed on producers, secondary consumers feed on primary consumers, and so forth. Energy is lost as heat at each transfer, following the second law of thermodynamics, which limits the length of food chains and determines the structure of ecological communities. Trophic levels organize organisms according to their position in food webs, with each level representing a step in the transfer of energy through ecosystems. Primary producers form the first trophic level, herbivores comprise the second level, and carnivores occupy higher levels. Decomposers play a crucial role by breaking down dead organic matter and returning nutrients to the system. The pyramid structure of trophic levels reflects the decreasing amount of energy available at higher levels, which typically supports fewer and larger organisms. Nutrient cycling represents one of the most important ecosystem processes, ensuring that essential elements remain available for biological processes despite their finite supply. The carbon cycle connects atmospheric carbon dioxide with organic molecules in living organisms and carbon stored in soils, sediments, and fossil deposits. The nitrogen cycle involves complex transformations between different chemical forms of nitrogen, with bacteria playing crucial roles in nitrogen fixation, nitrification, and denitrification. The phosphorus cycle lacks a significant atmospheric component, making phosphorus availability a limiting factor in many ecosystems. Population dynamics describe how and why population sizes change over time, influenced by birth rates, death rates, immigration, and emigration. Exponential growth can occur when populations have unlimited resources and favorable conditions, but environmental resistance typically limits growth as populations approach the carrying capacity of their environment. Population regulation involves density-dependent factors that increase in strength as populations become denser, such as competition for resources, predation, and disease, as well as density-independent factors like natural disasters that affect populations regardless of their size. Species interactions create the complex relationships that structure ecological communities and influence the evolution of species characteristics. Competition occurs when organisms use the same limited resources, potentially leading to competitive exclusion if one species is significantly superior, or to resource partitioning if species can specialize on different aspects of the resource. Predation involves one organism consuming another, creating predator-prey dynamics that can influence population cycles and community structure. Herbivora represents a special form of predation where animals consume plants, often leading to plant defenses and herbivore counter-adaptations. Mutualistic relationships benefit both species involved and can be essential for survival and reproduction. Plant-pollinator mutualisms enable plants to reproduce while providing food for pollinators. Mycorrhizal associations between fungi and plant roots enhance nutrient uptake for plants while providing carbohydrates for fungi. Cleaning symbioses involve one species removing parasites from another, benefiting both partners. These positive interactions demonstrate that cooperation, as well as competition, shapes ecological communities. Parasitism represents a relationship where one species benefits at the expense of another, without immediately killing the host. Parasites have evolved remarkable adaptations for exploiting their hosts while avoiding host defenses. Host-parasite relationships can influence population dynamics and community structure, while also driving evolutionary arms races between parasites and their hosts. Understanding parasitism is important for medical and veterinary applications, as well as for ecological theory. Biodiversity encompasses the variety of life at genetic, species, and ecosystem levels, representing one of Earth's most important characteristics. Species diversity includes both the number of species in a community and their relative abundances, with high diversity generally associated with stable environmental conditions and complex habitat structure. Genetic diversity within species provides the raw material for adaptation and evolution. Ecosystem diversity encompasses the variety of habitat types and ecological processes across landscapes. High biodiversity generally enhances ecosystem stability and resilience, while providing numerous benefits to human societies. Succession describes predictable changes in community composition over time following disturbances, demonstrating the dynamic nature of ecological communities. Primary succession occurs in areas lacking soil, such as newly formed volcanic islands or retreating glaciers, beginning with pioneer species that can tolerate harsh conditions and gradually progressing to more complex communities. Secondary succession takes place in areas where soil remains after disturbances such as fires, storms, or human activities, typically proceeding more rapidly than primary succession due to the presence of soil and seed banks. Community structure reflects the result of species interactions, environmental conditions, and historical factors that determine which species coexist and their relative abundances. Keystone species have disproportionately large effects on community structure relative to their abundance, often by controlling the abundance of other species through predation or competition. Foundation species create habitat structure that supports many other species, while indicator species reflect environmental conditions and can signal ecosystem health. Understanding community structure helps predict how communities will respond to environmental changes. Habitat and niche concepts help explain how species coexist and partition resources within ecosystems. Habitat refers to the physical environment where organisms live, including both abiotic factors like temperature and rainfall, and biotic factors like food availability and predators. The ecological niche encompasses not only the physical habitat, but also the functional role of a species within its ecosystem, including its resource requirements, environmental tolerances, and interactions with other species. The competitive exclusion principle suggests that two species cannot occupy identical niches indefinitely. Biogeography examines the distribution of species and ecosystems across Earth's surface, revealing patterns that reflect both current environmental conditions and historical processes. Climate represents the most important factor determining large-scale patterns of species distribution, with temperature and precipitation creating major biome types. Geographic barriers such as mountains, oceans, and deserts can isolate populations and promote speciation. Island biogeography theory predicts species diversity on islands based on their size and isolation, with larger and less isolated islands supporting more species. Disturbance plays a crucial role in maintaining ecosystem diversity and function by creating spatial and temporal heterogeneity. Natural disturbances such as fires, storms, floods, and disease outbreaks have shaped ecosystems throughout evolutionary history, and many species have adaptations for surviving or even benefiting from disturbances. The intermediate disturbance hypothesis suggests that moderate levels of disturbance maintain the highest diversity by preventing competitive exclusion while not eliminating less competitive species. Understanding disturbance regimes is essential for ecosystem management and conservation. Human impacts on ecosystems have become so pervasive that many scientists consider the current era a new geological epoch, the Anthropocene. Habitat destruction and fragmentation represent the greatest threats to biodiversity worldwide, as natural areas are converted to human uses or divided into small isolated patches. Climate change is altering temperature and precipitation patterns globally, forcing species to adapt, migrate, or face extinction. Pollution in various forms degrades ecosystem health and function, while invasive species introduced by human activities can disrupt native ecosystems. Conservation biology applies ecological principles to protect biodiversity and maintain ecosystem function in the face of human impacts. Protected areas such as national parks and nature reserves provide refuges for species and preserve important habitats, though they must be large enough and well connected to maintain viable populations and ecological processes. Restoration ecology seeks to repair damaged ecosystems and return them to more natural states, requiring understanding of ecosystem structure and function. Sustainable resource management attempts to balance human needs with ecosystem conservation. Climate change represents one of the most pressing environmental challenges of our time, with widespread implications for ecosystems and human societies. Rising temperatures are shifting species distributions toward poles and higher elevations, while changing precipitation patterns are altering ecosystem structure and function. Ocean acidification, caused by increased atmospheric carbon dioxide, threatens marine ecosystems. Extreme weather events are becoming more frequent and severe, causing disturbances that exceed the adaptive capacity of many species and ecosystems. As human populations continue to grow and consume more resources, understanding ecological principles becomes increasingly important for sustainable development. Ecosystem services such as climate regulation, water purification, pollination, and nutrient cycling provide enormous benefits to human societies, yet these services are often taken for granted until they are lost. Recognizing the value of ecosystem services can help guide policy decisions and promote conservation. The future of human civilization depends on our ability to live within Earth's ecological limits while maintaining the natural systems that support all life.
Chapter 1: The Nature of Disease, From Health to Pathology. Disease represents a fundamental disruption of the biological processes that maintain life, transforming the orderly symphony of molecular interactions that characterize health into the discordant chaos of pathology. Understanding disease requires examining how normal biological functions become altered, leading to the clinical manifestations we recognize as illness. This exploration takes us from the molecular level, where protein misfolding or genetic mutations initiate pathological cascades, to the systemic level, where organ dysfunction creates the symptoms that bring patients to seek medical care. The concept of health itself provides the baseline against which disease must be understood. Health represents a dynamic state of physical, mental, and social well-being that extends beyond the mere absence of disease. At the cellular level, health requires the maintenance of homeostasis through countless regulatory mechanisms that keep cellular environments within narrow parameters compatible with life. Metabolic pathways must function efficiently to provide energy and building materials. DNA repair mechanisms must prevent the accumulation of harmful mutations, and immune surveillance must eliminate threats while avoiding autoimmune responses. The transition from health to disease often begins with subtle molecular perturbations that gradually amplify into clinically apparent pathology. A single amino acid substitution in hemoglobin creates the misshapen red blood cells of sickle cell anemia, while the gradual accumulation of misfolded proteins in neurons leads to the progressive degeneration seen in Alzheimer's disease. These examples illustrate how minor molecular changes can have profound consequences for human health, demonstrating the exquisite sensitivity of biological systems to disruption. Pathogenesis, the development and progression of disease, involves complex interactions between genetic predisposition, environmental factors, and host responses. Some diseases result primarily from genetic defects that directly impair cellular function, while others arise from environmental insults such as infections, toxins, or radiation. Most diseases represent the interplay of multiple factors, with genetic susceptibility determining how individuals respond to environmental challenges. Understanding these multifactorial interactions is crucial for developing effective prevention and treatment strategies. The cellular response to injury reveals fundamental mechanisms that can either restore health or contribute to disease progression. When cells encounter stress or damage, they activate various protective mechanisms, including heat shock proteins that refold damaged proteins, DNA repair systems that correct genetic errors, and antioxidant defenses that neutralize harmful reactive oxygen species. However, when these protective mechanisms are overwhelmed or fail, cells may undergo programmed cell death or apoptosis, which can be beneficial if it eliminates damaged cells but harmful if it leads to excessive tissue loss. Inflammation represents one of the most important biological responses to injury and a central component of many diseases. The inflammatory response involves a complex cascade of molecular signals that recruit immune cells to sites of damage, increase blood flow and vascular permeability, and activate tissue repair mechanisms. While acute inflammation serves protective functions by eliminating pathogens and promoting healing, chronic inflammation can become pathological, contributing to diseases ranging from atherosclerosis to cancer. The balance between beneficial and harmful inflammation often determines disease outcomes. The role of genetics in disease extends from rare single-gene disorders that follow Mendelian inheritance patterns to complex diseases influenced by multiple genetic variants. Monogenic diseases such as cystic fibrosis or Huntington's disease result from mutations in single genes with large effects, making their inheritance patterns predictable and their molecular mechanisms relatively straightforward to understand. In contrast, complex diseases like diabetes, heart disease, and schizophrenia involve multiple genes, each contributing small effects that interact with environmental factors in unpredictable ways. Environmental factors play crucial roles in disease development, often serving as triggers that activate genetic predispositions or as direct causes of tissue damage. Infectious agents represent one of the most important environmental causes of disease, ranging from viruses that hijack cellular machinery to bacteria that produce toxic metabolites. Chemical exposures can damage DNA, disrupt cellular membranes, or interfere with enzymatic processes. Physical agents, such as radiation or mechanical trauma, can directly damage tissues or trigger inflammatory responses that lead to secondary complications. The concept of disease susceptibility helps explain why some individuals develop diseases while others remain healthy, despite similar exposures. Genetic polymorphisms can influence how efficiently individuals metabolize toxins, respond to infections, or repair cellular damage. Age represents a universal susceptibility factor, as the accumulation of molecular damage over time gradually impairs cellular function and increases disease risk. Lifestyle factors such as diet, exercise, and stress levels can modify disease susceptibility by influencing gene expression, immune function, and cellular repair mechanisms. Disease classification systems attempt to organize the vast diversity of human pathology into meaningful categories that facilitate understanding, diagnosis, and treatment. Diseases can be classified by their underlying causes, with categories including genetic diseases, infectious diseases, autoimmune diseases, and neoplastic diseases. Alternative classification schemes focus on affected organ systems, disease duration, or pathological mechanisms. Each classification system provides different insights into disease biology while highlighting the interconnected nature of pathological processes. The temporal dynamics of disease development reveal important patterns that influence both understanding and treatment. Acute diseases develop rapidly and often have identifiable precipitating events, while chronic diseases evolve slowly over months or years through gradual accumulation of damage. Some diseases follow predictable courses with well-defined stages, while others exhibit unpredictable fluctuations between periods of stability and acute exacerbations. Understanding these temporal patterns helps clinicians predict disease progression and time interventions appropriately. Molecular biomarkers provide objective measures of disease processes that can aid in diagnosis, prognosis, and treatment monitoring. These biological indicators range from simple laboratory tests that measure enzyme levels or inflammatory markers to sophisticated genomic analyses that identify disease-associated genetic variants. Biomarkers can detect disease before symptoms appear, monitor treatment responses, or predict disease progression. The development of increasingly sensitive and specific biomarkers continues to revolutionize medical practice by enabling personalized approaches to disease management. The concept of disease heterogeneity recognizes that what appears to be a single disease entity often represents multiple distinct conditions with different underlying mechanisms. Cancer provides a striking example, with hundreds of different cancer types that share some common features but differ dramatically in their molecular drivers, clinical behavior, and treatment responses. Similarly, conditions like diabetes or autism spectrum disorders encompass diverse subtypes that require different therapeutic approaches. Understanding disease heterogeneity is essential for developing targeted treatments. Systems biology approaches to disease seek to understand pathology as emergent properties of complex biological networks rather than focusing on individual molecular defects. These approaches recognize that diseases often result from perturbations in regulatory networks that control cellular function, tissue organization, or organ system interactions. Network-based analyses can identify key nodes or pathways that serve as therapeutic targets and can predict how interventions might affect multiple interconnected processes. The relationship between disease and evolution provides important insights into human pathology and potential therapeutic approaches. Many disease susceptibility genes persist in human populations because they provided evolutionary advantages in ancestral environments, even though they increase disease risk in modern contexts. The sickle cell trait protects against malaria while causing sickle cell disease in homozygotes, illustrating how evolutionary trade-offs shape disease patterns. Understanding evolutionary perspectives on disease can inform prevention strategies and highlight potential therapeutic targets. Emerging concepts in disease biology continue to reshape our understanding of pathology and open new therapeutic possibilities. The recognition that many diseases involve dysfunction of cellular organelles, such as mitochondria or the endoplasmic reticulum, has led to new treatment approaches targeting these structures. The discovery of epigenetic mechanisms that can silence genes without altering DNA sequences has revealed new categories of disease mechanisms and potential therapeutic targets. Advances in understanding the human microbiome have shown how microbial communities influence health and disease in previously unrecognized ways. As our knowledge of disease biology continues to expand, the boundaries between different diseases become increasingly blurred, while common underlying mechanisms become more apparent. This deeper understanding enables the development of therapeutic strategies that target fundamental pathological processes rather than just treating symptoms. The future of medicine lies in leveraging this mechanistic understanding to develop personalized treatments that address the specific biological abnormalities underlying each individual's disease, moving beyond the one-size-fits-all approaches that have dominated medical practice throughout history. Chapter 2: Infectious Disease Biology. Infectious diseases result from the complex interplay between pathogenic microorganisms and their human hosts, representing an ongoing evolutionary arms race that has shaped both human biology and microbial evolution throughout history. These diseases demonstrate how microscopic organisms can dramatically alter human health by exploiting cellular machinery, evading immune responses, and disrupting normal physiological processes. Understanding infectious disease biology requires examining the diverse strategies that pathogens use to establish infection, the host defenses that attempt to eliminate them, and the dynamic interactions that determine disease outcomes. Pathogenic microorganisms encompass a remarkable diversity of life forms, each with unique mechanisms for causing disease. Bacteria represent some of the most successful pathogens, using strategies ranging from direct tissue invasion to the production of powerful toxins that disrupt cellular function. Streptococcus pneumoniae causes pneumonia by adhering to lung epithelial cells and triggering inflammatory responses that impair gas exchange, while Clostridium botulinum produces botulinum toxin that blocks neurotransmitter release and causes paralysis. The diversity of bacterial pathogenic mechanisms reflects millions of years of evolution that have produced highly specialized strategies for exploiting human biology. Viral pathogens represent the ultimate cellular parasites, lacking the metabolic machinery necessary for independent existence and therefore requiring host cells for replication. Viruses have evolved sophisticated mechanisms for entering cells, hijacking cellular resources, and evading immune recognition. Influenza viruses constantly mutate their surface proteins to escape antibody recognition, while HIV integrates its genetic material into host chromosomes and establishes persistent infections that are extremely difficult to eliminate. The rapid replication and high mutation rates of viruses enable them to adapt quickly to host defenses and therapeutic interventions. Fungal infections pose particular challenges because fungi are eukaryotic organisms that share many cellular features with human cells, making it difficult to develop treatments that selectively target pathogens without harming the host. Opportunistic fungal pathogens such as Candida species normally exist as harmless commensals but can cause serious infections when host immune defenses are compromised. Pathogenic fungi have evolved various virulence factors, including the ability to change morphology, produce adhesins that facilitate tissue invasion, and secrete enzymes that damage host tissues. Parasitic diseases demonstrate some of the most complex pathogen-host interactions, involving organisms that have evolved intricate life cycles that often require multiple host species. Plasmodium parasites that cause malaria have a complex life cycle involving both mosquito vectors and human hosts, with different developmental stages adapted to survive in different cellular environments. The parasite's ability to alter the surface proteins it presents to the immune system enables it to establish chronic infections that can persist for years despite active immune responses. The process of infection begins with pathogen transmission and establishment in the host, involving multiple steps that each represent potential targets for prevention or treatment. Pathogens must first encounter susceptible hosts through various transmission routes, including respiratory droplets, contaminated food or water, sexual contact, or vector-borne transmission. Once pathogens gain access to host tissues, they must adhere to specific cellular receptors, survive host defense mechanisms, and establish a replicative niche that supports their multiplication and spread. Microbial virulence factors represent the specific molecular mechanisms that enable pathogens to cause disease, ranging from toxins that directly damage host cells to sophisticated systems that manipulate host immune responses. Diphtheria toxin inhibits protein synthesis in host cells, leading to cell death and tissue necrosis. Cholera toxin activates cellular signaling pathways that cause massive fluid secretion in the intestine, leading to life-threatening dehydration. Some pathogens produce virulence factors that enhance their survival and transmission, such as the capsules that protect bacteria from phagocytosis or the enzymes that break down tissue barriers. Host-pathogen interactions involve dynamic molecular dialogues where both organisms attempt to gain advantage through various strategies. Pathogens evolve mechanisms to evade or subvert host immune responses, while hosts develop countermeasures to recognize and eliminate invading microorganisms. Some pathogens disguise themselves by molecular mimicry, producing proteins that resemble host molecules and thereby avoid immune recognition. Others actively suppress immune responses by producing immunosuppressive molecules or by infecting and destroying immune cells themselves. The human immune system has evolved multiple layers of defense against infectious agents, creating formidable barriers that pathogens must overcome to establish successful infections. Innate immunity provides immediate responses through physical barriers, antimicrobial peptides, complement proteins, and phagocytic cells that recognize common pathogen-associated molecular patterns. Adaptive immunity develops specific responses against individual pathogens through T and B lymphocytes that can remember previous encounters and mount rapid secondary responses upon re-exposure. Immune evasion strategies employed by successful pathogens reveal the ongoing evolutionary pressure between hosts and parasites. Antigenic variation enables pathogens to change their surface proteins and escape antibody recognition, as seen in influenza viruses and many bacterial pathogens. Some viruses establish latent infections in which they remain dormant in host cells until conditions favor reactivation, while others integrate into host genomes and persist indefinitely. Immunosuppression represents another strategy, with pathogens producing factors that directly inhibit immune cell function or redirect immune responses away from effective antimicrobial activities. The development of antimicrobial resistance represents one of the most serious challenges in infectious disease medicine, demonstrating evolution in action as pathogens adapt to therapeutic pressure. Bacteria can acquire resistance through spontaneous mutations, horizontal gene transfer, or the activation of dormant resistance genes. The widespread use of antibiotics creates strong selective pressure that favors resistant strains, leading to the emergence of multi-drug resistant organisms that pose serious clinical challenges. Understanding the molecular mechanisms of resistance is crucial for developing new therapeutic strategies and preserving the effectiveness of existing treatments. Emerging infectious diseases highlight the ongoing threat posed by novel pathogens and the factors that enable their appearance in human populations. Zoonotic diseases that jump from animal reservoirs to humans represent a major source of emerging infections, often facilitated by ecological changes, increased human-animal contact, or viral mutations that enhance human-to-human transmission. Climate change, deforestation, urbanization, and global travel all contribute to the emergence and spread of new infectious diseases, creating conditions that favor pathogen evolution and transmission. The role of the human microbiome in infectious disease reveals the complex ecological relationships between commensal microorganisms and potential pathogens. The normal microbial flora provides protection against infection by competing for nutrients and attachment sites, producing antimicrobial compounds and stimulating immune development. Disruption of the microbiome through antibiotic treatment or other factors can increase susceptibility to infections by opportunistic pathogens. Understanding these ecological relationships is leading to new therapeutic approaches, such as fecal microbiota transplantation and probiotic treatments. Vaccine development represents one of the greatest successes in infectious disease prevention, demonstrating how understanding pathogen biology can lead to effective interventions. Traditional vaccines use killed or attenuated pathogens to stimulate protective immune responses without causing disease. Modern vaccine strategies include subunit vaccines that use purified pathogen components, DNA vaccines that encode antigenic proteins, and viral vector vaccines that deliver pathogen genes using harmless carrier viruses. The development of effective vaccines requires understanding which pathogen components elicit protective immunity and how to present them to the immune system most effectively. Diagnostic approaches to infectious diseases have evolved from simple clinical observation to sophisticated molecular techniques that can identify pathogens with unprecedented speed and accuracy. Traditional culture methods remain important for many bacterial infections and provide opportunities for antimicrobial susceptibility testing. Rapid diagnostic tests enable point-of-care detection of many pathogens, while molecular techniques, such as polymerase chain reaction, can detect minute quantities of pathogen genetic material. Advanced sequencing technologies can identify unknown pathogens and track transmission patterns during disease outbreaks. Therapeutic strategies for infectious diseases must balance the goals of eliminating pathogens while minimizing harm to the host, often complicated by the close evolutionary relationship between human cells and many pathogens. Antibiotics target processes that are essential for bacterial survival but absent in human cells, such as cell wall synthesis or bacterial protein synthesis. Antiviral drugs often target viral enzymes or replication processes that differ from their host counterparts. The development of new therapeutic approaches requires understanding pathogen biology at the molecular level and identifying vulnerabilities that can be exploited without harming the host. The global burden of infectious diseases remains enormous, despite advances in prevention and treatment, with particular impact on developing countries where poverty, malnutrition, and limited healthcare access create conditions that favor disease transmission and progression. Neglected tropical diseases affect over 1 billion people worldwide, often causing chronic disability rather than acute mortality. Understanding the social, economic, and environmental factors that influence infectious disease patterns is crucial for developing effective public health interventions and achieving global health equity. Public health approaches to infectious disease control rely on understanding transmission dynamics and implementing interventions that break the chain of infection. Surveillance systems monitor disease patterns and detect outbreaks before they spread widely. Isolation and quarantine measures can prevent transmission from infected individuals, while contact tracing identifies potential exposures. Vector control programs target arthropod vectors that transmit diseases such as malaria and dengue fever. These population-level interventions complement individual-level treatments and prevention measures to reduce the overall burden of infectious disease. The future of infectious disease biology will likely be shaped by continuing pathogen evolution, changing environmental conditions, and advancing technologies for prevention, diagnosis, and treatment. Climate change may alter the geographic distribution of vector-borne diseases, while increasing global connectivity facilitates rapid pathogen spread. However, advances in genomic sequencing, immunology, and drug development provide new tools for combating infectious diseases. Understanding the fundamental biology of host-pathogen interactions will remain crucial for addressing both established and emerging infectious disease threats. Chapter 3: Cancer Biology and Neoplastic Disease. Cancer represents one of the most complex and devastating categories of human disease, arising from the fundamental breakdown of cellular growth control mechanisms that normally maintain tissue homeostasis. This disease demonstrates how the same biological processes that enable multicellular life, including cell division, differentiation, and programmed cell death, can become corrupted to produce malignant growths that threaten the entire organism. Understanding cancer biology requires examining how normal cells transform into cancer cells, how tumors grow and spread, and how the body's natural defenses can both prevent and inadvertently promote malignant progression. The hallmarks of cancer encompass the fundamental cellular capabilities that distinguish malignant cells from their normal counterparts, providing a framework for understanding the diverse manifestations of neoplastic disease. Cancer cells acquire the ability to sustain proliferative signaling by producing their own growth factors or becoming hypersensitive to growth-promoting signals. They evade growth suppressors by inactivating tumor suppressor genes that normally prevent inappropriate cell division. Resistance to cell death enables cancer cells to survive in conditions that would normally trigger apoptosis, while unlimited replicative potential allows them to divide indefinitely without the senescence that limits normal cells. The genetic basis of cancer involves the accumulation of mutations that disrupt normal cellular function, transforming cells through a multi-step process that typically requires several genetic alterations. Oncogenes represent normal genes that promote cell growth and division, but become cancer-promoting when mutated or overexpressed. Tumor suppressor genes normally prevent cancer by restraining cell division or promoting cell death when DNA damage is detected, but their inactivation removes these protective mechanisms. DNA repair genes maintain genomic stability, and their dysfunction leads to increased mutation rates that accelerate cancer development. Tumor suppressor genes illustrate how the loss of normal cellular functions can contribute to cancer development. The p53 protein, often called the guardian of the genome, responds to DNA damage by halting cell division and either promoting DNA repair or triggering cell death if the damage is too severe. Mutations in p53 occur in over half of all human cancers, allowing cells with damaged DNA to survive and proliferate. The retinoblastoma protein controls progression through the cell cycle, and its inactivation permits uncontrolled cell division. Understanding how tumor suppressor genes function and how their loss contributes to cancer has led to new therapeutic strategies. Oncogenes demonstrate how normal growth-promoting genes can become cancer-causing when their activity is enhanced through mutation, amplification, or chromosomal rearrangement. The ROS proteins relay growth signals from cell surface receptors to the nucleus, but mutated ROS proteins remain constitutively active, driving continuous proliferation signals. The MYC transcription factor promotes cell division and growth, but its overexpression in many cancers leads to uncontrolled proliferation. Growth factor receptors can become oncogenic when overexpressed or mutated to remain active without appropriate growth signals. The process of carcinogenesis involves the progressive accumulation of genetic alterations that gradually transform normal cells into increasingly malignant forms. This multi-step process typically begins with exposure to carcinogens that damage DNA and disrupt normal cellular function. Chemical carcinogens such as those found in tobacco smoke can directly modify DNA bases, while radiation can cause DNA strand breaks. Biological carcinogens, including certain viruses, can integrate into host genomes or produce proteins that interfere with normal cell cycle control. DNA repair mechanisms play crucial roles in preventing cancer by correcting the genetic damage that would otherwise lead to malignant transformation. Base excision repair removes modified DNA bases, while nucleotide excision repair eliminates bulky DNA lesions. Mismatch repair corrects errors that escape proofreading during DNA replication, and homologous recombination repairs double-strand breaks. Inherited defects in DNA repair genes create cancer predisposition syndromes that dramatically increase cancer risk, demonstrating the importance of genomic maintenance in cancer prevention. The tumor microenvironment encompasses the complex ecosystem of cells, signaling molecules, and extracellular matrix that surrounds cancer cells and profoundly influences their behavior. Cancer-associated fibroblasts produce growth factors and matrix proteins that support tumor growth, while tumor-associated macrophages can either promote or inhibit cancer progression, depending on their activation state. Blood vessels that supply tumors often have abnormal structure and function, creating hypoxic regions that promote aggressive cancer cell behavior and resistance to therapy. Metastasis represents the most deadly aspect of cancer, involving the complex process by which cancer cells spread from their original location to distant sites in the body. The metastatic cascade requires cancer cells to invade surrounding tissues, enter blood or lymphatic vessels, survive circulation, exit vessels at distant sites, and establish new tumor growth in foreign tissue environments. Each step presents significant challenges, and most cancer cells that attempt metastasis fail to successfully establish distant tumors. However, the few cells that complete this process can seed life-threatening metastasis. Angiogenesis, the formation of new blood vessels, represents a critical capability that tumors must acquire to grow beyond minimal sizes. The angiogenic switch occurs when the balance between pro- and anti-angiogenic factors shifts to favor new vessel formation. Tumor blood vessels differ from normal vessels in having irregular structure, increased permeability, and abnormal flow patterns. Understanding angiogenesis has led to the development of anti-angiogenic therapies that attempt to starve tumors by cutting off their blood supply. Cancer stem cells represent a subset of cancer cells that possess enhanced ability to initiate tumor growth, resist therapy, and drive tumor recurrence. These cells share characteristics with normal tissue stem cells, including the ability to self-renew and differentiate into multiple cell types. Cancer stem cells may be responsible for tumor initiation, progression, and therapeutic resistance, making them important targets for cancer therapy. The stem cell model of cancer suggests that effective treatments must eliminate cancer stem cells rather than just reducing tumor bulk. The immune system plays complex and sometimes contradictory roles in cancer development and progression. Immune surveillance mechanisms can recognize and eliminate cancer cells, particularly those displaying foreign antigens or stress signals. However, chronic inflammation can promote cancer development by creating tissue damage, stimulating cell proliferation, and producing growth factors. Tumors can evade immune responses through various mechanisms, including downregulating antigen presentation, recruiting immunosuppressive cells, and producing inhibitory factors that impair immune function. Hereditary cancer syndromes demonstrate how inherited genetic defects can dramatically increase cancer risk, providing insights into the genetic basis of cancer and opportunities for prevention. Lynch syndrome results from inherited mutations in DNA mismatch repair genes and predisposes to colorectal and other cancers. Hereditary breast and ovarian cancer syndrome involves mutations in BRCA1 and BRCA2 genes that normally repair DNA double-strand breaks. Li-Fraumeni syndrome involves p53 mutations and predisposes to multiple cancer types. Understanding these syndromes has led to genetic testing programs and preventive strategies for high-risk individuals. Viral oncogenesis demonstrates how certain viruses can cause cancer by interfering with normal cellular growth control mechanisms. Human papillomavirus produces proteins that inactivate p53 and retinoblastoma tumor suppressors, leading to cervical and other cancers. Epstein-Barr virus can immortalize B lymphocytes and contribute to lymphoma development. Hepatitis B and C viruses cause chronic liver inflammation that can lead to hepatocellular carcinoma. Understanding viral contributions to cancer has led to preventive vaccines and new therapeutic approaches. Cancer treatment strategies have evolved from primarily surgical and radiotherapeutic approaches to include sophisticated molecular therapies that target specific genetic alterations in cancer cells. Chemotherapy uses cytotoxic drugs that preferentially kill rapidly dividing cells, though these agents also affect normal tissues and cause significant side effects. Targeted therapy focuses on specific molecular abnormalities in cancer cells, such as kinase inhibitors that block growth factor signaling or antibodies that target overexpressed receptors. Immunotherapy harnesses the patient's immune system to attack cancer cells through approaches such as checkpoint inhibitors and adoptive cell transfer. Precision medicine approaches to cancer treatment use molecular profiling of individual tumors to guide therapeutic decisions, recognizing that cancers with similar appearances may have different underlying genetic drivers and treatment responses. Genomic sequencing can identify actionable mutations that predict response to specific targeted therapies. Tumor profiling may also reveal resistance mechanisms that explain why certain treatments fail. The goal of precision medicine is to match each patient with the most effective treatment based on the specific molecular characteristics of their cancer. Drug resistance represents a major challenge in cancer treatment, arising through various mechanisms that enable cancer cells to survive therapeutic interventions. Cancer cells can mutate drug targets to reduce therapy effectiveness, amplify drug targets to overcome inhibition, or activate alternative signaling pathways that bypass block targets. Efflux pumps can remove chemotherapy drugs from cells, while DNA repair mechanisms can overcome the effects of DNA-damaging agents. Understanding resistance mechanisms is leading to combination therapies that attack multiple targets simultaneously and strategies to prevent or overcome resistance. The heterogeneity of cancer poses significant challenges for both understanding and treating these diseases. Tumors typically contain diverse populations of cancer cells with different genetic alterations, growth characteristics, and treatment sensitivities. This intratumor heterogeneity means that treatments may eliminate some cancer cells while allowing resistant populations to survive and proliferate. Spatial heterogeneity refers to differences between cancer cells in different regions of the same tumor, while temporal heterogeneity describes how tumors evolve over time through continued mutation and selection. Cancer prevention strategies focus on reducing exposure to known carcinogens and promoting behaviors that reduce cancer risk. Tobacco cessation programs target the leading preventable cause of cancer, while vaccination against cancer-causing viruses can prevent infection-associated cancers. Screening programs can detect precancerous lesions or early cancers when they are most treatable. Understanding the biology of cancer development has identified numerous opportunities for primary prevention that could dramatically reduce the global burden of cancer. Emerging areas in cancer biology continue to reveal new aspects of malignant transformation and potential therapeutic targets. The role of metabolism in cancer has gained renewed attention, with recognition that cancer cells often have altered metabolic requirements that could be exploited therapeutically. Epigenetic alterations that change gene expression without altering DNA sequences represent another active area of research and drug development. The contribution of mechanical forces and tissue architecture to cancer development is leading to new understanding of how physical factors influence tumor progression. Chapter 4: Autoimmune and Inflammatory Diseases. Autoimmune and inflammatory diseases represent a diverse group of conditions in which the immune system, normally responsible for protecting the body against foreign threats, becomes misdirected against the body's own tissues or fails to properly regulate inflammatory responses. These diseases illustrate the delicate balance required for immune system function and demonstrate how the same mechanisms that provide protection against infections can become pathological when dysregulated. Understanding these conditions requires examining the complex interplay between genetic susceptibility, environmental triggers, and immune system dysfunction that leads to chronic inflammation and tissue damage. The immune system operates through intricate networks of cellular and molecular interactions designed to distinguish self from non-self and eliminate threats while avoiding damage to healthy tissues. This remarkable system includes innate immunity that provides immediate responses to danger signals, and adaptive immunity that develops specific responses against particular antigens. Regulatory mechanisms including suppressor T cells, anti-inflammatory cytokines, and checkpoint molecules normally prevent excessive immune responses and maintain immune tolerance to self-antigens. When these regulatory systems fail, autoimmune diseases can develop. Autoimmune diseases arise when the immune system loses tolerance to self-antigens and begins attacking the body's own tissues as if they were foreign invaders. This breakdown of self-tolerance can occur through various mechanisms including molecular mimicry, where pathogens share structural similarities with self-antigens and trigger cross-reactive immune responses. Epitope spreading can expand autoimmune responses as tissue damage releases additional self-antigens that become targets for immune attack. Bystander activation can stimulate autoreactive immune cells in the context of inflammation caused by infections or other triggers. The genetic basis of autoimmune diseases involves complex interactions between multiple susceptibility genes and environmental factors. Human leukocyte antigen (HLA) genes, which encode proteins that present antigens to T cells, show strong associations with many autoimmune diseases. Certain HLA alleles may present self-antigens in ways that promote autoimmune responses, while others may be protective. Non-HLA genes affecting immune regulation, cytokine production, and apoptosis also contribute to autoimmune disease susceptibility. However, genetic factors alone are insufficient to cause autoimmune diseases, indicating the importance of environmental triggers. Environmental factors play crucial roles in triggering autoimmune diseases in genetically susceptible individuals. Infections represent important triggers through various mechanisms, including molecular mimicry, bystander activation, and adjuvant effects that enhance immune responses. Chemical exposures can modify self-proteins to create new antigens, or directly damage tissues to release sequestered antigens. Physical trauma can similarly release hidden antigens and trigger autoimmune responses. Stress, diet, and other lifestyle factors may also influence autoimmune disease development through effects on immune function and inflammation. Type 1 diabetes exemplifies organ-specific autoimmune disease, where immune responses target a specific cell type, resulting in the destruction of insulin-producing beta cells in the pancreas. The disease involves both cellular and humoral immune responses, with T cells directly attacking beta cells and antibodies serving as markers of the autoimmune process. Environmental triggers, such as viral infections, may initiate the autoimmune response in genetically susceptible individuals. The progressive destruction of beta cells leads to absolute insulin deficiency and the characteristic metabolic abnormalities of diabetes. Rheumatoid arthritis represents systemic autoimmune disease characterized by chronic inflammation of synovial joints that can lead to progressive joint destruction and disability. The disease involves complex interactions between genetic factors, environmental triggers, and immune system dysfunction. Autoantibodies against citrullinated proteins and rheumatoid factor serve as diagnostic markers and may contribute to disease pathogenesis. Inflammatory cytokines, including tumor necrosis factor and interleukin-6, drive synovial inflammation and joint destruction, making them important therapeutic targets. Multiple sclerosis demonstrates how autoimmune responses can target the central nervous system, leading to demyelination and neurological dysfunction. The disease appears to involve T-cell-mediated immune responses against myelin proteins, though the specific antigens and triggering events remain incompletely understood. The relapsing-remitting course typical of early disease suggests episodic immune activation, while the progressive phases may reflect ongoing neurodegeneration. Environmental factors including viral infections, vitamin D deficiency, and geographic latitude influence disease risk and progression. Systemic lupus erythematosus illustrates how autoimmune responses can affect multiple organ systems simultaneously, creating diverse clinical manifestations that can be difficult to diagnose and treat. The disease involves the production of autoantibodies against nuclear antigens, including DNA and histones, leading to immune complex formation and deposition in various tissues. These immune complexes trigger complement activation and inflammatory responses that can damage kidneys, joints, skin, and other organs. The episodic nature of lupus flares suggests that environmental factors periodically trigger increased autoimmune activity. Inflammatory bowel diseases, including Crohn's disease and ulcerative colitis, demonstrate how immune responses against the normal intestinal microbiota can lead to chronic intestinal inflammation. These diseases involve complex interactions between genetic susceptibility, environmental factors, the intestinal microbiome, and mucosal immune responses. Normally, the intestinal immune system maintains tolerance to beneficial microorganisms while remaining capable of responding to pathogens. In inflammatory bowel disease, this balance is disrupted, leading to inappropriate inflammatory responses against the normal microbiota. The role of regulatory T cells in maintaining immune tolerance and preventing autoimmune disease has become increasingly recognized as these specialized immune cells suppress potentially harmful immune responses. Regulatory T cells express the transcription factor FOXP3 and produce anti-inflammatory cytokines that inhibit effector immune responses. Defects in regulatory T cell development or function can lead to autoimmune diseases, while enhancing regulatory T cell activity represents a potential therapeutic strategy. The balance between effector and regulatory immune responses often determines whether autoimmune diseases develop or remain in remission. Molecular mimicry represents an important mechanism by which infections can trigger autoimmune diseases through cross-reactive immune responses between pathogen and self-antigens. Rheumatic fever provides a classic example, where immune responses against streptococcal antigens cross-react with cardiac proteins and can cause carditis. Guillain-Barré syndrome can follow infections with Campylobacter jejuni due to antibodies that cross-react with nerve gangliosides. Understanding molecular mimicry has implications for vaccine development and may explain why certain infections are associated with increased autoimmune disease risk. Chronic inflammation plays central roles in many diseases beyond classical autoimmune conditions, contributing to atherosclerosis, metabolic syndrome, neurodegenerative diseases, and cancer. This chronic inflammatory state often involves the same cytokines and cellular mediators seen in autoimmune diseases, but may arise through different mechanisms, including tissue damage, metabolic dysfunction, or aging-related cellular stress. The recognition that inflammation contributes to diverse diseases has led to investigation of anti-inflammatory therapies for conditions not traditionally considered inflammatory. The complement system, a cascade of proteins that can directly kill pathogens and enhance inflammatory responses, plays important roles in both protective immunity and autoimmune disease pathogenesis. Complement activation can clear immune complexes and apoptotic cells, helping to prevent autoimmune responses. However, excessive complement activation can also cause tissue damage and promote inflammation. Complement deficiencies can predispose to both infections and autoimmune diseases, while complement inhibitors represent promising therapies for certain autoimmune conditions. Therapeutic approaches to autoimmune and inflammatory diseases have evolved from non-specific immunosuppression to targeted therapies that interfere with specific inflammatory pathways. Corticosteroids remain important for controlling acute inflammation, but have significant side effects with long-term use. Disease-modifying anti-rheumatic drugs can slow disease progression in conditions like rheumatoid arthritis. Biological therapies, including antibodies against inflammatory cytokines, have revolutionized treatment of many autoimmune diseases. Small molecule inhibitors of intracellular signaling pathways offer additional therapeutic options. The concept of immune privilege helps explain why certain tissues such as the eyes and brain are less susceptible to autoimmune attack through specialized mechanisms that limit immune responses. These tissues express factors that inhibit immune cell activation and may lack conventional lymphatic drainage that would expose antigens to the immune system. However, immune privilege can be broken under certain conditions, leading to autoimmune diseases such as autoimmune uveitis or multiple sclerosis. Understanding immune privilege may suggest strategies for protecting tissues from autoimmune attack. Tolerance induction represents the ultimate therapeutic goal for autoimmune diseases, aiming to re-establish immune tolerance to self-antigens and eliminate the need for chronic immunosuppression. Experimental approaches include administering autoantigens in tolerogenic forms, using regulatory T-cell therapy, and modifying antigen-presenting cells to promote tolerance rather than immunity. While successful tolerance induction remains elusive for most human autoimmune diseases, continued research may eventually make this goal achievable. Personalized medicine approaches to autoimmune diseases use biomarkers to predict disease course, treatment responses, and potential side effects. Genetic testing can identify individuals at high risk for certain autoimmune diseases or particular drug toxicities. Cytokine profiles may predict which patients will respond to specific biological therapies. Monitoring autoantibody levels can help assess disease activity and treatment responses. As our understanding of autoimmune disease heterogeneity improves, more precise therapeutic approaches will become possible. The burden of autoimmune and inflammatory diseases continues to increase in developed countries, possibly due to reduced microbial exposures in early life that fail to properly educate the developing immune system. The hygiene hypothesis suggests that decreased exposure to infectious agents may increase autoimmune disease risk by allowing immune systems to become hypersensitive to self-antigens. Understanding the factors that promote healthy immune development may suggest prevention strategies for autoimmune diseases. Prevention strategies for autoimmune diseases remain limited due to incomplete understanding of disease triggers and the complex interactions between genetic and environmental factors. However, some interventions show promise, including vitamin D supplementation for multiple sclerosis prevention and elimination of gluten for celiac disease prevention in susceptible individuals. As knowledge of autoimmune disease mechanisms improves, more effective prevention strategies may become available. The future of autoimmune and inflammatory disease research will likely focus on understanding the fundamental mechanisms that maintain immune tolerance and how these mechanisms fail in disease. Advances in genomics, proteomics, and single-cell analysis are revealing new details about immune system function and dysfunction. These insights will guide the development of more precise therapies that can restore immune balance without compromising protective immunity. The ultimate goal remains the development of curative treatments that can permanently restore immune tolerance and eliminate the need for lifelong immunosuppression. Chapter 5: Genetic Diseases and Molecular Medicine. Genetic diseases arise from alterations in DNA sequences that disrupt normal gene function, demonstrating how information encoded in our genes directly influences health and disease. These conditions range from rare disorders caused by mutations in single genes to complex diseases influenced by multiple genetic variants interacting with environmental factors. Understanding genetic diseases requires examining how genes control cellular function, how mutations alter these processes, and how genetic information can be used to diagnose, treat, and prevent disease. The field of molecular medicine applies this knowledge to develop new therapeutic approaches that target the fundamental molecular defects underlying genetic disorders. The molecular basis of genetic diseases reflects the central role of genes in controlling cellular function through their encoded proteins. Genes provide instructions for synthesizing proteins that serve as enzymes, structural components, transport molecules, and regulatory factors essential for life. When mutations alter gene sequences, the resulting proteins may be non-functional, have altered activity, or be produced in abnormal quantities. These protein defects then cascade through cellular and physiological systems to produce the clinical manifestations of genetic disease. Single gene disorders follow Mendelian inheritance patterns and result from mutations in individual genes with large effects on protein function. Autosomal recessive disorders, such as cystic fibrosis and sickle cell anemia, require 2 copies of mutated genes for disease expression, often resulting from consanguineous marriages or founder effects in isolated populations. Autosomal dominant disorders, including Huntington's disease and familial hypercholesterolemia, require only 1 mutated copy for disease expression and typically show vertical transmission through families. X-linked disorders, such as hemophilia and Duchenne muscular dystrophy, predominantly affect males due to their single X chromosome. Cystic fibrosis exemplifies how single gene defects can have multi-system consequences through disruption of fundamental cellular processes. The disease results from mutations in the CFTR gene that encodes a chloride channel essential for normal ion transport across epithelial cells. Loss of CFTR function leads to thick, sticky secretions that obstruct airways and digestive ducts, causing progressive lung disease, pancreatic insufficiency, and other complications. The most common mutation, deletion of phenylalanine at position 508, causes the protein to misfold and be degraded before reaching the cell surface. Sickle cell disease demonstrates how even single amino acid changes can have profound consequences for protein function and human health. The disease results from a mutation in the beta-globin gene that substitutes valine for glutamic acid at position 6, causing hemoglobin molecules to polymerize under low oxygen conditions. This polymerization distorts red blood cells into characteristic sickle shapes that can block blood vessels and cause painful crises, organ damage, and shortened lifespan. The same mutation provides protection against malaria in heterozygous carriers, illustrating how genetic variants can have both beneficial and harmful effects. Huntington's disease illustrates how expansion of DNA repeat sequences can cause neurodegenerative diseases through toxic protein aggregation. The disease involves expansion of CAG trinucleotide repeats in the Huntington gene, leading to production of proteins with extended polyglutamine tracks. These abnormal proteins aggregate in neurons and cause progressive neurodegeneration affecting movement, cognition, and behavior. The number of repeats correlates with age of onset, with longer expansions causing earlier disease manifestation, demonstrating how genetic variation can influence disease severity and progression. Mitochondrial diseases represent a unique category of genetic disorders caused by mutations in mitochondrial DNA, which is maternally inherited and encodes essential components of the cellular energy production machinery. These diseases typically affect tissues with high energy demands, such as brain, heart, and skeletal muscle. Mitochondrial DNA mutations can cause various clinical syndromes, including mitochondrial encephalomyopathy, Leber hereditary optic neuropathy, and mitochondrial cardiomyopathy. The maternal inheritance pattern and variable clinical manifestations reflect the unique biology of mitochondrial genetics. Chromosomal disorders result from abnormalities in chromosome number or structure that can affect multiple genes simultaneously. Down syndrome, caused by trisomy 21, demonstrates how an extra chromosome can cause intellectual disability and multiple congenital anomalies through gene dosage effects. Turner syndrome results from complete or partial absence of one X chromosome in females, leading to short stature, ovarian dysgenesis, and other features. Chromosomal rearrangements, such as deletions, duplications, and translocations, can disrupt gene function through various mechanisms, including gene disruption, position effects, and altered gene dosage. Complex genetic diseases involve multiple genes, each contributing small effects that interact with environmental factors to influence disease risk. Type 2 diabetes, coronary artery disease, and schizophrenia represent complex diseases where numerous genetic variants combine with lifestyle factors to determine individual disease susceptibility. Genome-wide association studies have identified many genetic variants associated with complex diseases, though these variants typically explain only small portions of disease heritability. Understanding complex disease genetics requires consideration of gene-gene interactions, epigenetic modifications, and environmental influences. Pharmacogenomics examines how genetic variations influence drug responses, enabling personalized medicine approaches that optimize therapeutic effectiveness while minimizing adverse reactions. Genetic polymorphisms in drug metabolizing enzymes can dramatically affect how quickly individuals clear medications from their systems, influencing both efficacy and toxicity. Variations in drug targets can determine whether medications will be effective, while polymorphisms in drug transporters can affect tissue distribution. Understanding pharmacogenomic relationships enables clinicians to select optimal medications and dosages based on patients' genetic profiles. Genetic testing technologies have evolved from simple family history analysis to sophisticated molecular techniques that can detect various types of genetic alterations. Chromosomal analysis can identify large-scale abnormalities, while DNA sequencing can detect single nucleotide changes. Array-based technologies can simultaneously test for thousands of genetic variants associated with disease risk. Whole exome and whole genome sequencing provide comprehensive genetic information, but require sophisticated interpretation to distinguish disease-causing variants from benign polymorphisms. Gene therapy represents the ultimate application of molecular medicine, aiming to treat genetic diseases by introducing functional genes into patients' cells. Various approaches include replacing defective genes with normal copies, supplementing gene function, or silencing disease-causing genes. Vector systems including modified viruses, lipid nanoparticles, and physical methods enable gene delivery to specific tissues. Success stories include treatments for severe combined immunodeficiency, inherited blindness, and hemophilia, though challenges remain in achieving tissue-specific delivery and long-term gene expression. Genome editing technologies such as CRISPR-Cas9 enable precise modification of DNA sequences and offer potential cures for genetic diseases through correction of disease-causing mutations. These tools can delete, insert, or modify specific DNA sequences with unprecedented precision and efficiency. Applications include correcting mutations that cause sickle cell disease, treating inherited blindness, and potentially preventing genetic diseases through germline editing. However, genome editing raises important ethical considerations regarding safety, equity, and the appropriate limits of genetic modification. Newborn screening programs demonstrate how understanding genetic disease mechanisms can enable early detection and prevention of serious complications. Screening for phenylketonuria enables dietary interventions that prevent intellectual disability, while detection of sickle cell disease allows prophylactic treatments that reduce complications. Expanded screening panels now test for dozens of genetic conditions, though implementation requires careful consideration of test accuracy, treatment availability, and potential psychological impacts on families. Genetic counseling provides essential support for individuals and families dealing with genetic diseases, helping them understand inheritance patterns, risk assessments, and available options for testing and treatment. Genetic counselors work with patients to interpret genetic test results, assess reproductive risks, and make informed decisions about medical care. Prenatal genetic counseling can help couples understand risks to their offspring and available options for testing and intervention. The expanding capabilities of genetic testing require skilled genetic counselors to help patients navigate increasingly complex information. Preimplantation genetic diagnosis enables couples at risk for genetic diseases to select embryos free of disease-causing mutations during in vitro fertilization procedures. This approach can prevent transmission of serious genetic diseases while avoiding the need for pregnancy termination following prenatal diagnosis. However, preimplantation genetic diagnosis raises ethical questions about embryo selection and the potential for enhancement rather than just disease prevention. The technology continues to evolve with improved genetic testing methods and expanded applications. Personalized medicine approaches use genetic information to tailor medical care to individual patients, recognizing that genetic variations influence disease risk, progression, and treatment responses. Cancer treatment increasingly relies on molecular profiling of tumors to guide therapeutic decisions, while pharmacogenomic testing helps optimize drug selection and dosing. Polygenic risk scores attempt to predict complex disease risk based on multiple genetic variants, though their clinical utility remains limited. The goal of personalized medicine is to provide the right treatment to the right patient at the right time, based on their unique genetic and molecular characteristics. Ethical considerations surrounding genetic diseases and molecular medicine include issues of privacy, discrimination, equity, and the appropriate use of genetic information. Genetic discrimination in employment and insurance remains a concern despite legal protections, while the high cost of genetic tests and treatments raises questions about healthcare equity. The ability to edit human embryos raises fundamental questions about human enhancement and the potential consequences for future generations. Balancing the benefits of genetic knowledge with protection of individual rights requires ongoing dialogue between scientists, clinicians, ethicists, and society. The future of genetic diseases and molecular medicine will likely be shaped by continuing advances in genetic technologies, computational biology, and our understanding of gene function. Artificial intelligence approaches may improve interpretation of genetic variants and prediction of disease outcomes. Single-cell technologies will provide new insights into how genetic mutations affect cellular function. Advanced gene therapy and genome editing approaches may enable treatment of previously incurable genetic diseases. As these technologies mature, the challenge will be ensuring equitable access to their benefits while addressing ethical concerns about genetic modification and enhancement. Emerging concepts in genetic disease biology continue to reveal new mechanisms of pathogenesis and potential therapeutic targets. The recognition that many genetic diseases involve disruption of cellular organelles, such as lysosomes, mitochondria, or the endoplasmic reticulum, has led to new treatment approaches targeting these structures. Epigenetic modifications that alter gene expression without changing DNA sequences represent another category of genetic alterations that may be more amenable to therapeutic intervention than traditional mutations. Understanding how genetic variants interact with environmental factors will be crucial for developing comprehensive approaches to genetic disease prevention and treatment.